Okay, so chapter 7 is all about membrane structure and function. We talked a little bit about membranes in the last chapter when we went through the cell, and talked about them when we talked about lipids back in chapter 5. So we know that the phospholipids are what make up the membranes, and we know that the membrane is responsible for controlling what comes into and what gets out of the cell. So we'll talk about that in more detail as we go through this chapter. So let's review the phospholipid structure. So if you remember, the phospholipid consists of a phosphate group, a glycerol backbone, and those two long chain fatty acids. And these can either be saturated fatty acids, like the one shown here, or they can be unsaturated, which have the double bonds and therefore the kinks. So this is the general structure of the phospholipid. Remember that the head groups with that phosphate group, they are going to be polar or hydrophilic. So these guys will, as we know, dissolve in water. And then these tails, the long fatty acid chain tails, are going to be nonpolar or hydrophobic, and so they're going to avoid water or not be dissolvable in water. Okay, so the cell membrane, remember, it controls what comes into and what goes out of the cell, and it is what we call selectively permeable. That means it allows some things to easily pass through the membrane and other things not so easily pass through the membrane. So things like gases, water, and small non-polar molecules can easily diffuse across the membrane. And these non-polar molecules are key because remember, most of the structure of the membrane is non-polar. And remember, we learned back in chapter 3 that like dissolves like. So polar things like to interact with other polar things, and non-polar things like to interact with other non-polar things. So non-polar molecules will easily cross through this majority non-polar cell membrane. Now, this X here is a little bit tricky, so when I put that X there, I don't mean to imply that glucose and large polar molecules and ions absolutely do not cross the cell membrane. It just means that they don't do it easily. So they do not easily cross the cell membrane. But yes, they can indeed cross, usually with the help of things like pumps or channels or something else that's going to help those molecules get across the cell membrane. All right, so the model of the cell membrane is what we call a fluid mosaic model. So this means that fluid means that every membrane is different. So we have a mixed composition, meaning that it's not always the same phospholipids, it's not always the same proteins. So it can vary from organism to organism and even from tissue to tissue. Now, there are these specialized proteins embedded in the phospholipid bilayer, and we'll talk about the different types of proteins and what those are. But we do see the total composition of a cell membrane consists of the lipid bilayer, plus the proteins, the specialized proteins, and we'll talk about what they are. So the other aspect of this that sort of makes up the fluid part is that the membranes are flexible, they can drift, and they are not rigid. Okay, so they can spin. So the fatty acids that make up the lipid bilayer are they're fluid, they move, they're not rigid, they're not stiff, but they can bend, they can flex, they can spin, and so that makes up sort of the fluid aspect of the fluid mosaic model. All right, so let's talk about what some of those proteins are, those specialized proteins that are embedded in the membrane. There are 4 main classes of proteins. We have the adhesion proteins, the transport proteins, the recognition proteins, and the receptor proteins. We're going to talk a little bit about each one of these on the next slides. All right, so adhesion proteins help cells of the same type locate each other. So remember, we talked about the term adhesion when we talked about water, and basically that means things sticking to something else. And in this case, we're talking about cell types that stick together and that stick to the proper tissue. So for example, they stick to the chest cavity if they're cardiac muscles or cardiac cells, or they stick to the liver tissue if they're liver cells, or they stick to the pancreas tissue if they're pancreatic cells. So these adhesion proteins help cells of the same type kind of locate each other, find each other, stick together, and remain in the proper tissue. So that's an adhesion protein. A transport protein is obviously, that name sort of is self-explanatory. These are proteins that are there to help molecules move across the membrane. So remember, we talked about things like glucose and ions and large polar molecules not being able to easily diffuse across the cell membrane. Well, they need these transport proteins in order to help them get across. So an example might be this GLUT1 protein, and that is responsible for transporting glucose inside your cells. The third type of specialized protein that we want to talk about are the recognition proteins. This is what basically identifies self from non-self. So every cell in your body has a special, unique identity marker on it that designates it as your cell. If a bacteria or a virus enters your body, it would not have that specific marker, and that is the way that your immune cells recognize that cell as non-self and something that we need to attack. So the recognition proteins are there to identify self from non-self. And then lastly, we have the receptor proteins. So receptor proteins are what allows us to respond to cues in our extracellular environment and to regulate the cellular responses. So there are all kinds of signals and cues that our cells are constantly being bombarded with, and the receptor proteins that are present on the membrane have to respond to those cues, or have to detect those cues and then respond to those cues. All right, so we talked about this term diffusion a little bit already as we talked about water and gases and things like that diffusing across the cell membrane. So I want to talk a little bit about this idea of diffusion. So when we talk about diffusion, we mean net movement of molecules or ions, and we say that that occurs down a concentration gradient. When we refer to a concentration gradient, we're talking about the difference in the number of molecules or ions in different regions. So for example, in this bowl here, we have a concentration gradient of food dye. We have a very high concentration gradient of dye here in this area. And over here in this area, so this would be a high concentration. Over here, where we have no colored particles, that would be a low concentration gradient. So as those dye particles start to spread out inside the beaker, we get something that looks like this second picture here. Now we still have a concentration gradient here because we have a high area of concentration over here, and kind of over here in the beaker or in the flask, we still have a low concentration. So diffusion continues. So we get the third flask or the third beaker here that looks like this. And you can see here we no longer have a concentration gradient. There is no difference. We can't pick a region where there's more colored dye particles and fewer colored dye particles. They are evenly distributed throughout the water, and so we no longer have a concentration gradient. So basically, when things diffuse, you get movement of the ions or molecules down the concentration gradient from a region of high concentration to a region of low concentration, and that happens until we reach what is called equilibrium. So in this third beaker, we have reached equilibrium, and that is when there is no net movement of molecules. So the molecules are evenly distributed, and even though the molecules still move slightly, there's no net movement of the molecules. So molecules move from what we call an area of high concentration to an area of low concentration, and that's always going to be true. Molecules are always going to move from a highly concentrated area to a low concentrated area, always. All right, so there are three factors that affect or influence the rates of diffusion. One we just kind of talked about is the concentration gradient. So the greater the gradient, the more distinction we have in the number of molecules from one area to the next, the faster the rate of diffusion is going to occur. And again, that's going to occur until we reach equilibrium. The other thing that affects the rates of diffusion are molecular size. So small molecules are obviously going to move faster than larger molecules. And then lastly, we have temperature. So the higher the temperature, the faster the faster the molecules will move, and the faster the rate of diffusion will occur. So three things that affect the rate and direction of diffusion. Okay, so when we're talking about diffusion of solutes across the cell membrane, and by solutes we're talking about small nonpolar molecules or water, we can have two types of transport that can occur. The first is called passive transport, and this is it's also sometimes called facilitated diffusion. That's just another name for passive transport. So this is where substances again move down that concentration gradient, so an area of high concentration to an area of low concentration. Passive diffusion does not require any energy. So remember, ATP is our energy currency. Passive diffusion does not require ATP. The other type of transport is called active transport, and this is where you get movement of substances against a concentration gradient. So this would be from an area of low concentration to an area of high concentration. So this type of transport does require energy. So in order to get those molecules to move against the concentration gradient, we have to supply some energy. There are two examples of this. They're called endocytosis and exocytosis that we're going to talk about. First, I want to talk about facilitated diffusion or passive transport. So again, these are molecules that are moving down the concentration gradient, so areas of high concentration to areas of low concentration, and no ATP is required, or no energy is required. So we do not, my pen stopped working. We do not require ATP here, okay, so no ATP. So if you look here, we have our cell membrane here. We have our extracellular space, so that's outside the cell membrane. And then we have our intracellular space down here, that's inside the cell membrane. So here you can see extracellularly, we have a high concentration of these ions or molecules or whatever it is that we want to move across the cell membrane. So we have a channel here. So this is just a pore in the membrane that is going to allow these molecules to pass through. So you can see they can just pass right through that channel or that pore. They can go in, they can go out, depending on the channel. Sometimes they can be bidirectional, so they can move both ways depending on the gradient. But in this case, we are primarily going to have molecules flowing in to the cell because of the way the concentration gradient is set up. We are high extracellularly and we are low intracellularly. So without that ATP, we are going to go down the concentration gradient, and molecules are going to move from the area of high concentration to the area of low concentration. Now, with active transport, we can have movement of molecules against the concentration gradient. So this would be going from again, areas of low concentration to areas of high concentration. It does require ATP, and usually the molecules are pumped through protein. So here we have both a sodium pump and a potassium pump examples, and you can see that we are requiring ATP here in order to move these molecules through. So they enter the pump, we have to break down some ATP, and then so ATP is used here, and then the molecules exit the pump on the other side. So this would be, again, movement of molecules from an area of low concentration to an area of high concentration. We'll talk about when that would be important a little bit later on. All right, so then the two other examples of active transport are endocytosis and exocytosis. So endocytosis is when we are trying to move a large group of molecules inside the cell. So this is movement inside a cell. Okay, so if we're talking endocytosis, we are moving things inside. So here, this is extracellular out here or outside the cell, and this is inside down here in this kind of beigey area. So you can see here we have a large group of molecules here that we want to move inside the cell. So what happens is you get sort of this pouch that forms on this is the cell membrane. So the cell membrane sort of bunches up and forms this little pouch that can then sort of be pinched off right here along this dotted line, and we end up with a vesicle that contains all of the molecules that we wanted to move inside the cell. So now once they're inside the cell, they can break out and they can do whatever it is we need them to do inside the cell. So this is ATP requiring. It does require ATP in order to do this, but it's an effective strategy when we want to move a whole bunch of molecules across the cell. Exocytosis is kind of the opposite. So here we are moving things out of the cell. So again, here up top we have our extracellular environment, and down here we have inside the cell, or the intracellular environment. So here we already have all of our molecules inside this vesicle. So again, that's surrounded by the cell membrane. They're already in the vesicle. The vesicle is going to move up to the plasma membrane. So here's our plasma membrane. I'll label it PM. The vesicle is going to situate itself right up next to that plasma membrane, and then it's going to fuse with the membrane and become part of the membrane. And as it does that, it expels all of these molecules outside the cell. Again, this is an ATP requiring process, so it falls into the active transport category. Okay, so we've talked a lot about moving ions or molecules across the membrane, but we also know that water is one of those substances that easily moves across the membrane, and we need to talk about how water flows across the membrane. It's so important that we give it its own special term, which we refer to as osmosis. So this is diffusion of water, again, down, water's going to be, it's diffusion of water due to a water concentration gradient between two regions that are separated by this membrane. Now again, water, just like everything else, is going to move down the concentration gradient. So again, without the use of ATP or energy to tell us otherwise, water is going to move from an area of high concentration to an area of low concentration. So the key here with water, what makes it a little bit tricky, is that it depends on the concentration of the solutes. So if you think about it, we rarely talk about the concentration of water. We talk about the concentration of solutes inside or dissolved in the water. Okay, so we have to kind of think about this in a relative way. So if we have two beakers, set up here, and we have solutes in one. So we have 12 solutes. So let's just call this 12 molar. And we have, over here we have 5 solutes. So we'll call this 5 molar. So beaker number 1 has what we call high solute. And again, this is relative to and compared to beaker number 2. It has high solute, so it would have low water. Beaker number 2 has low solute or low solute, and it would have then high water. All right, so when we're thinking about the concentration of water, we're always doing so relative to the concentration of solute. Okay, so let's look at this again with another example. And this is something similar to what you're going to be doing in lab. So when you do this in lab, it'll help this make a little bit more sense to you, or give you a reference point or example. So here we have 2 reservoirs separated by a selectively permeable membrane. So water as well as solute molecules can move across this membrane. Remember that the net movement of water molecule is always from a high concentration to a low concentration until we obtain equilibrium. And that's the key, right? Until the water concentration and the solute concentration is balanced out, we're going to have movement. So you can see over here we have all these little green solute molecules. So this side is very high solute concentration, therefore it's going to be low water. This other side is exactly the opposite. We have no green molecules over here, so we have a very low solute concentration over here, and therefore we have high water. So again, we're going to get movement of water from an area of high concentration to an area of low concentration. We'll have the solutes moving this way to the left because they're high solutes over here, low solutes over here. And we're going to get movement back and forth across this membrane of water and solutes until we reach equilibrium, until we get even concentrations of water and solutes on both sides. Okay, so when we talk about the extent and direction of water movement across the membrane, we always compare 2 solutions. So we compare solution 1 to solution 2. So you're always talking about this in relative terms. You would never talk about water movement when you're just talking about one solution. So here we have 2 solutions. We have a beaker with 2 molar sucrose. A bag with 2 molar sucrose. And we have a beaker that has 0.5 molar sucrose. So 2 is higher than 0.5. So we would say that the bag has high solute, and therefore it would have low water or lower water compared to the beaker. The beaker would have low solute and therefore high water. So if we take this bag and we put it into our beaker of water, what's going to happen is water is going to move into this bag, causing the bag to swell, because water again is going to move from an area of high concentration, the beaker, to an area of low concentration, the bag. Now the terminology that we use here is the solution that has the high solute we refer to as hypertonic. The solution that has low solute we refer to as hypotonic. So you can kind of think about this, that if you have hypothermia, that means that your temperature is very low, so that refers to low solute. If you have a hyperactive child, you've got a child that's got a lot of energy or very high energy level, so that refers to the high solute concentration in that solution. So we can look at another example here where again we have our bag with 2 molar sucrose, but now in our beaker we have 10 molar sucrose. So comparatively, now our beaker is high solute, low water, and our bag is now low solute, high water. So now it is our beaker that is, excuse me, it's our beaker that is hypertonic, and it is our bag with the low solute that is hypotonic. All right, so now again, we're talking about water. Water, if we were to take this bag and put it into the beaker, water is going to move from an area of high concentration, which in this case is the bag, to an area of low concentration, which in this case is the beaker. So water is going to leave the bag, go out into the beaker, and that's going to cause that bag to shrivel. All right, we can also have a situation where we have what are called isotonic solutions. So here we have 2 molar sucrose in the bag and 2 molar sucrose in the beaker. So these solutions are isotonic. We could not say that one is hyper and one is hypo depending on the solute concentrations because they're the same. Now this does not mean we don't get movement of water. We do. The same amount of water that goes into the bag is going to go out of the bag. So water, there's going to be no net movement, but there is movement. So water's going into the bag, water's going out of the bag at the same rate and to the same extent. I'm sorry. It's no net movement of the water. So again, the rate at which water is entering the bag is equal to the rate at which water is leaving the bag. So a little bit about these, just refresh your memory on hypertonic and hypotonic. So hypotonic refers to the solution with fewer solutes, so low solute like hypothermia. Hypertonic refers to the solution with more solute or high solute, so like a hyperactive child. And one thing you can remember, if it helps you, is that water always moves from a hypotonic to a hypertonic. So remember, again, with the isotonic, that means we have no net movement of water, so we still have movement, but we don't have net movement. So here's just an example of some blood cells as they are placed in different solutions. You can see the blood cells in the hypotonic solution look kind of swollen and puffy, and because of all the water that's flowing into those cells. You can have red blood cells that were placed in a hypertonic solution that you can see are sort of starting to shrivel because of all the water exiting those cells. And then you see healthy red blood cells that have sort of the little dimple in the middle, as red blood cells do, in an isotonic solution. So again, here we have normal cells because the movement of water is the same into and out of the cells. All right, so that's the end of chapter 7. I will see you in class.